I'm Daniel Levine, and this is the Bio Report. RION is developing platelet-derived exosome therapeutics as off-the-shelf, room-temperature stable alternatives to traditional cell therapies. The company's lead program is advancing toward phase 3 trials in diabetic foot ulcers, and it's building a broader pipeline across musculoskeletal, cardiovascular, pulmonary, dermatologic, and women's health indications. The company's built on stem cell research performed at the Mayo Clinic that showed that the regenerative benefits from these treatments came from exosomes' biological signaling that promoted healing rather than transplanted stem cells themselves. We spoke to Atta Behfar, co-founder and CEO of RION, about how the company's purified exosome products work, their potential as scalable and cost-effective regenerative therapies, and how they avoid immune issues that have long hampered cell-based approaches. Atta, thanks for joining us.
Thank you so much for having me.
We're going to talk about exosomes, RION, and the company's efforts to use these to develop regenerative therapies that can overcome the limitations of cell-based therapies. Let's start with the exosomes themselves, though. Can you explain what these are?
Yeah, absolutely. So exosomes were actually initially described several decades ago, and they were thought to be cellular waste. So they were essentially an ignored byproduct of cell function for many, many years. But over the last 2 decades, they've increasingly been recognized as having a critical role in the normal functioning of cellular and actually organism processes. So your body really relies on exosomes for normal function. And so what are they? They are the modality by which a cell communicates with its environment. And essentially, the way I always describe it is that we use words to communicate with each other. Cells use exosomes. And the behavior of exosomes functions very much like words. In other words, when I speak, you hear the word and then it's gone. And an exosome, when it's absorbed into the recipient cell, it releases its payload, the message is delivered, and again, it disappears.
How varied are exosomes within the body, and how varied is the cargo they carry?
Extraordinarily varied. So just as we have numerous different types of cells in our bodies, you have the same number, or actually many more exosomes from each of these cells. So as these cells want to shed different messages into their environment, they will actually make different types of extracellular vesicles or exosomes. And each cell wants to say and do something differently in the body. And so each cell produces different exosomes, and there are, millions of different cells in the body, and each of them produce different ones. So you can imagine that there's an exponentially varied number of exosomes that are floating around your body at any given time.
We've discussed exosomes in the past on this podcast because there are some companies that are exploring them as vectors to deliver therapeutic payloads. I understand RION is looking at them for that purpose in the case of some of its preclinical candidates, but you're also pursuing platelet-derived exosomes because of their regenerative powers. What makes platelet-derived exosomes compelling as a therapy?
So it's a it's sort of a long story, how we came up with platelets as the source of exosomes. And I'll try to give you the Cliff Notes version of how we ended up here. Essentially, our program has been a stem cell program since the late 90s. This is our program out of Mayo Clinic, and then later on the Van Cleve Cardiac Regenerative Medicine program. And our focus was really to try and optimize stem cells that are adult-derived, so either bone marrow-derived or fat-derived stem cells, for their use in cardiac regeneration. And as we were working to develop this ideal cellular therapeutic phenotype, we came to realize that the cells themselves had much less to do with the benefit that was conferred to either preclinical models or even in the clinical setting, and rather what is being shed seems to be the most important. And what's being shed is the exosome. So the messages from the stem cells seem to be very important rather than the cells themselves being delivered. So that was step one, realizing that exosomes were very important in regeneration. Step two was then, well, how do we actually scale this in a way where we have a uniform product? And stem cells were the obvious choice, right? We're a stem cell lab. Why aren't we using stem cells for this? As we found out, and this is work that really took about 5 years, as we found out, when you take cells that divide, so cells that have a nucleus and they're able to self-renew, and you put them in a petri dish and you give them a nutrition to divide, as you expect them to do that outside the body, you very rapidly accumulate DNA damage. It's because these cells aren't designed to be in the ambient environment, nor are they designed to be outside of the pressures that you have, holding you together inside your body. And so when you put them in these environments where you're sort of whipping them to divide faster, they accrue DNA damage. And so we could never get a consistent exosome product from cultured stem cells. Additionally, the more we studied these very effective stem cells, these stem cells that seem to be very good at regeneration, the more we kept developing a very unique regenerative signature that looked like platelet signatures. Every time, we had in our culture systems, we had platelets and PRP, we had platelet lysate as controls. And when we were looking at, what makes stem cells effective, we kept seeing a lot of overlap with platelets. And we came to realize that these stem cells were really recycling the platelet content from the culture medium. And that was what was making them effective. So not only were the stem cells a bad model to manufacture the exosomes, as it turns out, the most regenerative exosome was coming from the platelet. Now, a platelet is something that isn't a cell, actually. It's a little piece of a megakaryocyte. There's recent papers that would categorize it as something called a blebosome, like a big chunk of a cell that's released. Doesn't have a nucleus, it can't divide, and it can only do a few things in the body, right? It's there, it's programmed to either go down road A, B, or C. Let's say A is regeneration. And so now our task was, well, can we take platelets, put them in an environment where we're assured that they're only going to go down road A, and then collect their exosomes, see what happens. And that's exactly what we did. And in doing so, we were able to get an extremely consistent regenerative exosome that now had all of the power of the most effective stem cell without any of the variability, dramatically reduced costs, dramatically increased scalability. And once we achieved that is when we really started up RION out of Mayo Clinic to now, invest dollars into it and be able to take it into practice.
That's so interesting. I'm wondering what advantages these might have over cell-based therapies.
So I bear the scar of the stem cell era and cell-based therapies over my younger career, let's say. So cell-based therapy, number one, cost. The cost of stem cell therapy or cell-based therapy is just astronomical. The amount of logistical infrastructure and actually disposable costs that it takes to simply have a single dose is huge. And so accessibility of that therapeutic to a broader population becomes limited, right? Because now you're looking at a product that may cost, in excess of $100,000 a dose. So that was number one. Number two is variability, right? So each donor, if you have a dividing cell, is going to provide a different cellular phenotype, right? You and I look different. Our cells look different. And so their behavior is going to be different. And so intrinsically, you're going to have different cellular function depending on the cellular source. Whether it's autologous or allogeneic, you're always going to have that difference. On top of that, you have a superimposition of the thing I said before, where now you're taking these cells out of their natural environment, putting them into an artificial environment, and then whipping them to divide. They're not going to be a very uniform cellular phenotype. So all in all, you have a high cost, high variability technology versus now something that we can massively scale at a very cost efficient manufacturing, from a manufacturing perspective, something that's very cost efficient. And logistically, we actually lyophilize these exosomes, meaning we freeze dry them. So they're stable at room temperature. On the stem cell side, you have to cryopreserve them. And so as they're being shipped to the recipient hospital, you would need to now have all the infrastructure to receive the cells, keep them frozen, thaw the cells out, ensure that they're all alive, prep them so you get rid of all the cryopreservation solution, concentrate them down, and then deliver the dose to the patient. So, like versus a freeze-dried vial where you just add saline and it's ready to go. So logistically, just a dramatic change in terms of how we can utilize them in practice.
You have a pipeline of therapies that combine what you call PEP or purified exosome product with other agents. What does it take to make the PEP? Do you start with donated blood?
Absolutely right. So what we have is a proprietary process that leverages platelets and essentially forces platelets into a regenerative state. And then we collect these regenerative exosomes off of the platelets as they are conditioning their environment in that very specific state. So that's PEP. Uh, if you think about it in your own experience, anytime you injure yourself, you see a bruise or you see blood, right? Um, the thing that you use to heal yourself, especially with soft tissue injury, are your platelets. So naturally, these platelets in your body are there to institute soft tissue healing. What we're doing is offering that message in conditions where the individual now has a disrupted regenerative function. In other words, you have an individual that's older, an individual that's peripheral vascular disease, diabetes, radiation injury, et cetera, et cetera, et cetera. We are now able to exogenously deliver the messages from those platelets in a manner that they no longer can achieve. And so we're simply replacing what would naturally occur in a healthy young individual.
And how scalable is your production, and does sourcing from blood create any supply constraint?
It actually is much more plentiful than any other cellular source. We are able to leverage the platelet supply chain in a way that does not disrupt access to platelets from a transfusion perspective. So we actually take platelets sort of at the end of their transfusibility. So we're not robbing the blood banking system of any transfusible blood products. And on top of that, because there's a shelf life to these platelets, they're actually pretty abundantly available. So we're able to leverage that resource, and the blood banks love it because if they have a home for these near expiration platelets, then they're much more willing to maintain a larger supply of platelets for transfusion. So it actually is a very symbiotic relationship.
And are there ways to grow or expand the exosome population once they're isolated, or is that just irrelevant?
You can't, you cannot grow exosomes or platelets after they've been isolated out of the body. So, and actually, that's the beauty of the system. We do not want self-proliferating cells because that's when you get accumulation of DNA damage. We want these to sort of be sort of at their end state in terms of biological function so that we ensure a high degree of reproducibility.
And they're allergenic. You can use this as an off-the-shelf product to treat anyone.
Indeed, you can use this and that's how we position it as a allogeneic resource. And what's very nice about regenerative platelet exosomes, I'm a heart transplant doctor, and we actually analyzed whether they have self, non-self markers on their surface and have observed, and this is in a couple of our publications and in our FDA filings, we've observed that they're devoid of those. So really they have immune privilege as they're being shed into the environment.
Your lead experimental therapy is for diabetic foot ulcers. What is it, and how does it work?
So in diabetes, you have a whole host of biological functions that start to not work anymore, right? So you have microvascular disease, you have peripheral neuropathy, and you have a disruption in normal cell growth at the level of your tissues. Here, what we do is we exogenously put the message of healing back into an area, into the ulcer, that is non-healing. And by delivering the exosomes into that local environment, it's as though we're massively upregulating the blood flow to that environment in terms of messaging. And what that does is actually cause the body to form new microvasculature. It causes the skin cells to proliferate. It causes the inflammation in the local environment to be subdued. And it creates a protective environment where cells that are sort of on the brink of cell death are resuscitated and actually can participate in tissue healing. So all those four processes get activated in that local environment, which then fosters a much more robust healing endeavor than what you would get with current standards of care.
And what's known about it from studies conducted to date?
So we definitively, and the data's embargoed because we're obviously under peer reviewed and things like that. But I can say that we do see a pretty dramatic increase in the healing of these. We've seen that in the phase 1 studies and also now in these phase 2 studies that do have a control arm as well as a treatment arm. And we're now essentially positioned to go down the pivotal trial arm.
So what would be the development path forward?
So we've met with the FDA. They have the phase 2 information. They've cleared the path for us to move forward with phase 3. Obviously, in the phase 3 effort, you want to have all your ducks in a row. In other words, you would like to have all your manufacturing to now be positioned commercially, so it's all compliant with what's called a BLA filing, or with sort of what you would take to market. And so we have all of the blueprint from the FDA as to the things they would like to see from us from a trial design and from a manufacturing perspective. These are all things that we're just tweaking, and we anticipate launching into the phase 3 effort in 26.
This has very broad potentials. You're looking at musculoskeletal, cardiovascular, pulmonary indications. Why start with diabetic foot ulcers?
For a couple of reasons. Number one, exosomes are, from a therapeutic perspective and a therapeutic class, they're new. Really, we're one of the unique companies that's been able to scale to a point where the exosome product is actually commercially viable. And strategically, what we endeavored to do was to go from outside in other words, to gain comfort with it both clinically and from a regulatory perspective. It would make sense to go after topical problems and then work our way inside the body so that we had a big safety dossier and also clinical recognition of safety for our providers. That's number one. Number two is much more, I would say, personal to me and many of my colleagues who are day in and day out taking care of patients. One of the biggest challenges we have in medicine is indeed non-healing wounds. It is an area that is the bane of our existence, and it is an area that has been really ignored from a biotherapeutic or small molecule development arena. Really, most of the products we have access to are something called devices that are approved under a different pathway at the FDA known as the 510K pathway. But true therapeutic, randomized controlled trials showing benefit is really lacking since the late 90s for wound healing. And so because our product is cost efficient, is accessible, it's mass producible, we felt very compelled to address this truly unmet need in medicine. And when I say unmet need, I'm talking in the United States, there's in excess of several million people annually, several million with non-healing wounds, right? So we're talking about 1% to 2% of the US population, right? So 1 in 100 to 1 in 50 that has this problem, where there's been not a drug approved in the last 25, 30 years. So it is an area that I feel is very ripe for innovation. And in all frankness, this stuff really works for it. So we ought to use it.
This is a topical treatment, but how do you deliver it for other indications? Can it be delivered systemically?
Depends on what you're trying to do. So for our musculoskeletal application, for example, we're doing something called intra-articular delivery, where we actually go into the joint space. We have preclinical data that does show not only do we get a decrement in inflammation. In other words, we alleviate the inflammatory environment that you see with degenerative joint disease. But we actually foster a healthy immune system in that local environment. But the preclinical data also suggests that we're getting both chondral, which means cartilage, and subchondral, which is the bone under the cartilage, regeneration. And so that was really the primer to go for intra-articular delivery. We also have data on tendon regeneration that we've published on. And so the next chapter, and we're working actually with our colleagues in the Department of Defense to stand up a trial where we're treating tendinopathies or diseases of the tendon with the exosome product. And we obviously have a big pipeline, as you alluded to, whereby we are now looking at this product for restoration of cardiovascular function, primarily in the realm of ischemic cardiomyopathy, meaning diseases where blood flow is lacking to the heart, whether that's heart attack or more chronic heart disease where there is a lack of blood flow. We're also looking at it for peripheral vascular disease, meaning you have lack of blood flow to your limbs. And moving forward, we have several additional endeavors where we're looking at other inflammatory and soft tissue indications, such as women's health application, as well as inflammatory bowel disease applications. And that's more down the road.
I know you've been able to access some non-dilutive funding, but how much has the company raised to date?
We have raised about $100 million to date. And with that, we've been able to really, in a highly efficient manner, get to pivotal trials and scale the product, which I think, if you compare us to other exosome efforts, is a pretty cost-efficient endeavor.
And how far will existing cash take you, and what's the plan for raising additional capital?
So the way that we've structured this company is that we've built a core team as almost a hub, whereby we have all the core expertise living under RION as a structure. What we then have done is built teams that are focused on different applications of the exosome product. One of the earliest applications was actually development of a product that we called human platelet extract that we use in the cosmetic world. And that company is already revenue generating. And so that helps both RION and that vertical out of RION really move forward in a unique way. Additionally, we've created a veterinary focused company where we're aiming to treat both horses and dogs with regenerative exosomes. We've created a company that is now focused on wound healing, as well as focused on inflammatory dermatology. And we're going to continue to create these spokes out of RION. So what happens is that as these spokes become successful, RION offers shared goods and services to these spokes. And so we're able to now operate without needing to raise more capital in the hub company. And we focus more on these verticals as the area where more capital is raised.
I'm curious whether you've seen a shift in investors' receptivity these days and what conversations with investors have been like.
So really, I think our focus has been to be as efficient as we can be in terms of our regulatory process and our trial process. And we convey those efficiencies with different investors. I would say that different foundations and different investors are very focused on different targets. You talk to somebody and they may say, "Oh, this is all really interesting. Are you doing anything in the eye? We're only going to invest in eye." So I think, you have investors that really focus their dollars to an area of expertise that they've built. And so what we're aiming to do is really to identify those investors that share our vision and the critical need for wound healing innovation and raise capital in wound healing. And additional investors may be interested in dermatology, but not necessarily wound healing. And those may be more focused on this I&I or inflammatory skin conditions, of which you probably see advertisements for on TV a lot. So it's a big area of continued innovation because it's a big area of need. And similarly for cardiovascular, for veterinary, for aesthetics applications, there are different investors that'll have those areas of focus.
Atta Behfar, co-founder and CEO of RION. Atta, thanks so much for your time today.
Thank you so much. Really appreciate your time.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.